Over the course of our three major macromolecules that we use through catabolism to produce ATP, we're going to be filling out this flowchart. This is something we'll actively do in lecture. However, we will start with this for carbohydrates. As we eat, we start the process of digestion in our mouth, specifically for our carbohydrates. More specifically for our starches of our carbohydrates. So digestion, or the saccharification, the action of breaking down saccharides, starts in the mouth with the enzyme amylase that can be found in our saliva. So looking at our carbohydrates first. So these first two boxes are from that big flow chart. And then this is going to be the middle box of this flow chart. Looking at our carbohydrates or our saccharides, we're going to really actually follow polysaccharides like amylose or amylopectin. The energy we get, if we're talking about food calories, is 4 calories per gram of our carbohydrates. And we're going to compare that to the energy we can get from proteins and from our lipids. The first process of digestion with amylase is the process called hydrolysis. Hydrolysis is where we are going to have water, hydro, used to split lysis, these glycosidic linkages. Our saliva is mostly made up of water, but we are going to have enzymes in there. I'm just going to give you the broad category of what these enzymes are called. And amylase is very specific to break down amylose. However, that's not the only carbohydrate enzyme we have. So this class of enzymes are called glycosidases. The ACE in this name is referring to the enzymes. So the glycosid means the enzymes that break down glycosidic linkages. And ultimately, what we are going to make are the monosaccharides. In this video, we're going to look more closely at how this process happens. Getting into the nitty-gritty. Okay, not that gritty, but let's see how this happens. Amylase enzymes break the glycosidic linkages in the presence of water. Because, remember, water was released through the condensation reaction of bringing two glucose molecules together. We need to then put that water back in to reform the two hydroxyl groups on either monosaccharide. Throughout this process, within our saliva, we can get a variety of different products. So depending on the enzyme used, will help us predict the products that come through this process of using amylases within our saliva. Here in this chart, we have our alpha amylase. So our alpha amylase is an enzyme that will randomly break alpha 1,4 glycosidic linkages. The product from this, because it is random, is the production of oligosaccharides. Oligosaccharides consist of about 3 to 15 monosaccharides or glucose molecules together. Those do serve other roles, or they can be further digested. The glucosamylase will specifically hydrolyze one alpha-1,4 or 1-alpha-1,6 glycosidic linkage. So here is where the branching can get broken up in like amylopectin. Because it's breaking one linkage at a time, it's going to release glucose as that product. The last type of enzyme is our beta amylase. Our beta amylases will break the alpha-1,4 glycosidic linkage. In particular, it's going to break it into a disaccharide. And since these are alpha-1,4 glycosidic linkages, the disaccharide being released specifically is maltose. I'm not going to test you on these different types of enzymes and what they do, but what I want you to understand is enzymes are present. They each have a role and a job, which is why we get a variety of different products out. This is the process starting in our mouth, through with our saliva, at the beginning of this digestive process.